Plastic-eating bacteria, a promising solution to plastic pollution. Plastic-eating bacteria have become a topic of great interest in recent years due to the increasing problem of plastic waste in the environment. Plastic pollution is affecting our planet, and traditional disposal and recycling methods are not enough to tackle the problem. This has led scientists to search for new solutions to the plastic waste crisis. In this regard, plastic-eating bacteria have emerged as a promising solution. What are plastic-eating bacteria? Plastic-eating bacteria are microorganisms capable of breaking plastic down into simpler components. They do this by secreting enzymes that can degrade the plastic into smaller units, making it easier to break down. These bacteria belong to the genus Ideanella and are known for their ability to break down and utilize polyethylene terephthalate (PET) as a food source. The first well-known plastic-eating bacteria is called Pseudomonas aeruginosa. This bacterium can break down polyurethane, a type of plastic commonly used in the production of foam products. Researchers discovered that the bacterium could produce an enzyme called polyurethane hydrolase, which breaks down the polyurethane polymer into smaller, biodegradable compounds. This process is known as biodegradation. Another plastic-eating bacteria is called Ideanella sakaiasis. This bacterium can break down polyethylene terephthalate (PET), which is commonly used in producing plastic bottles. The bacterium can produce an enzyme which breaks down the PET polymer into smaller, biodegradable compounds. A third well-known plastic-eating bacteria is called Bacillus subtilis. This bacterium is commonly found in soil and can break down polystyrene, a type of plastic widely used in producing foam products. Researchers have discovered that the bacterium can produce an enzyme called polystyrene depolymerase, which breaks down the polystyrene polymer into smaller, biodegradable compounds. The process of plastic degradation by bacteria starts with the attachment of the bacteria to the plastic surface. The bacteria then secrete enzymes that break down the plastic into smaller units, and it requires the presence of moisture, oxygen, and warmth. The primary enzyme used by plastic-eating bacteria is called PETase. This enzyme breaks down the plastic into two smaller units, monoethylene glycol and terephthalic acid. The bacteria can then use the MEG as a food source while the TPA is excreted. This process takes approximately 6 weeks, and the bacteria can continue to break down more plastic as long as the conditions are suitable. Using plastic-eating bacteria as a solution to plastic waste has several advantages over traditional methods. Firstly, the bacteria are efficient at breaking down plastic, reducing the time and energy needed to dispose of the waste. Secondly, the process is environmentally friendly, as it does not release harmful chemicals into the environment. Thirdly, the process is cost-effective, as the bacteria can be cultured in large numbers and used to break down plastic in a controlled environment. Additionally, the end product of the degradation process is biodegradable, which means it will not persist in the environment for centuries like traditional plastic. Although plastic-eating bacteria have many advantages, several challenges still need to be addressed. Firstly, the bacteria are still in the early stages of development and require further research to understand their potential applications. Secondly, plastic degradation is slow, and the bacteria need specific conditions to thrive, such as warmth, moisture, and oxygen. Thirdly, there are concerns about the scalability of the process, as the large-scale cultivation of the bacteria is challenging. Finally, the cost of producing the bacteria and the enzymes they secrete is still relatively high, making it challenging to implement the process on a large scale. In summary, plastic-eating bacteria have the potential to be a game-changer in the fight against plastic pollution. The bacteria are efficient, environmentally friendly, and cost-effective, making them a promising solution. However, further research is needed to fully understand these bacteria, potential and overcome the challenges they face. Suppose we can isolate and cultivate these bacteria and scale up the process. In that case, we could have a powerful tool for reducing the impact of plastic pollution on the environment. The use of these bacteria can revolutionize how we manage plastic waste, reducing its impact on the environment and helping to protect our planet for future generations. Thanks for watching.